Okay. Hello, everyone. Good morning, good afternoon, and good evening, wherever you are. Perhaps starting the seminar today without comment about the ongoing struggle for racial justice in the United States would provide a welcome break from what is going on. But for so many of our Black and Brown sisters and brothers, that privilege of being able to simply turn attention elsewhere does not exist. So I'm going to make just a brief statement here. I've spent a lot of time in self-reflection these past 2 weeks, taking a hard look at what efforts I've made in the past to try to promote social justice in my community and in my country. And in doing so, I have been confronted with this inescapable feeling of embarrassment and shame because of the inadequacy of my previous efforts. Growing up surrounded by conservative peers, I thought it was enough to behave better than they did. I have come to accept, though, perhaps embarrassingly late, that it was never enough to simply avoid harmful practices if it meant that I also remained silent while racist practices and ideas were perpetuated around me. It wasn't enough to be not racist. I mean, we're all called to be anti-racist. So if you're like me and you're feeling embarrassed, don't let that embarrassment turn into defensiveness. Instead, harness it and turn it into action. We can't change what we did or failed to do yesterday or last year or last decade, but we can choose what we do today and we can choose what we do tomorrow. And we're all called to use our voices, our privilege, our bodies, our time, or our money to advance the cause of justice. So if you've been fighting this struggle while I and so many others have remained silent, thank you for your efforts. And if you're in a similar position to me, don't allow feelings of past failure prevent you from reconciling with yourself and with your community and taking steps to move us forward towards a more just world. Welcome to the 5th week in the long-term animal research seminar series. We continue this week with Dr. Jacinta Beehner. And before I introduce Jacinta, I'll make a couple of announcements. First, if you're participating via Zoom, you should see a Q&A tab on the top or bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. At the end of the talk, we'll go through those questions, starting with the ones with the most votes. But if you have a clarifying question that you feel like needs to be addressed during the talk, in order for you to understand something, you can type clarification in capital letters at the start of your question. And Jacinta has said she'll do her best to answer those questions in real time. Second, recordings of all the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, this talk will be available for viewing and reviewing after it's complete. Now I'm very pleased to introduce today's speaker, Dr. Jacinta Beehner, professor in the departments of psychology and anthropology at the University of Michigan. Before becoming a professor, Jacinta received her bachelor's degree from Boston College and her PhD from Washington University at St. Louis. She then had 2 really productive and influential postdoc positions, first with Dorothy Cheney and Robert Seyfarth at the University of Pennsylvania, and then with Gene Altman at Princeton. Jacinta now directs 2 different long-term studies of wild primates, the Capuchins at Toboga project, and the project that we'll be hearing about today, the Simeon Mountains Gelada research project. Jacinta has made major contributions to our understanding in a number of areas, including life history theory, reproductive strategies, sexual conflict, and behavioral endocrinology. And what's so impressive to me is how Jacinta's work seamlessly connects ultimate and proximate mechanisms. Her work draws a path from endocrinology and other physiological mechanisms up through organismal level outcomes and all the way to population level processes. In addition to primary data production and analysis, she's also phenomenal at using review type articles to push her fields forward. And if you're at all interested in reproductive suppression or the links between glucocorticoids and fitness, do yourself a favor and go read her masterful reviews on those topics. So I'm really excited to welcome Jacinta now and hear about male-mediated changes in female reproduction.
Thanks so much, Matt. That was a very nice introduction. And I just really want to thank everyone for joining today, everyone who's able to join live. And I also want to thank everyone who might be hearing this recording later. Hopefully it's because you're all out fighting for social justice And I do just want to echo what Matt said. For any of us in positions of power, we really do need to harness that power like never before to make a change in this world. And it's not just something for this moment. Oh, last week I marched, this week I'll donate. It's really it's a commitment. It's really a commitment to change that really should continue until we actually see structural changes in this country. Okay, so also, before I begin, I'd like to take just another minute to thank my incredible team of collaborators for all the work that I'm going to present on today. The work on the Bruce effect really wouldn't have been possible without my former I'm focusing on deceptive swellings first. It wouldn't have been possible without a postdoc at Stony Brook University, Dr. Alice Benyal. And this is work in progress, and I'll be presenting on some of it today. The work on the Bruce effect wouldn't have been possible without my former graduate student, Dr. Ayla Roberts, who's now faculty at Michigan State University. And then the work on the Vandenberg effect and male-mediated maturation was catapulted forward largely due to the efforts of a current graduate student at Stony Brook University, Jacob Fetter. And of course, all of the whole Simeon Mountains Gelada project wouldn't even exist without Tori Bergman, who helped me co-found the project 15 years ago. He collaborates with me on everything from watching geladas, watching capuchins, raising children. And while the Gelada project wouldn't exist without Tori, it might not still exist without Amy and Noah. So Dr. Amy Liu and Dr. Noah Snyder-Mackler have joined on as co-directors. And as we're all learning throughout this great seminar series, long-term research is a marathon, not a sprint. So I'm just so grateful to have Amy and Noah, not just as co-directors and collaborators, but also just as really good friends. And that's what makes collaborating with them so incredibly fun and rewarding. Okay, so when Matt asked me to talk about long-term research in animal behavior, I knew I had just the talk. And especially since the final little piece of this story really was 15 years in the making. Now, this doesn't mean that any of the stories that I'm about to tell you couldn't use another 15 years of data to fill in some of the puzzles. But there comes a time in all research when it's just time to publish. And sometimes you just have to leave some questions open for now. So what I'd like to do today is to pull together 3 different lines of my research that all share a common theme. Each one presents an example of a male-mediated change in female reproduction in a wild gelada. As you'll see, they also seem to, they sort of share a common mechanism. And this is something I'll return to at the end of the talk. So I study geladas. Geladas are Cercopithecine primates, and they're close relatives of baboons, but they're not baboons. They're an entirely different genus, so don't call them gelada baboons. And the research I'm presenting today really began the first month of what now adds up to 15 years of research. On January 31st, 2006, all photos have dates on them now, so it's very easy to date them. I watched my first male takeover. I even snapped this photo. This is when I used to just snap photos for fun. And I think it's fitting that I start the talk with a photo from my first month. So what's going on here? Well, geladas live in polygynous groups where, for the most part, there's one dominant male, and he has mating rights to all the other females in the group. On the periphery of the group are these bachelor males, and they're constantly looking for any chance to upend the dominant male so that they can take over the group of females. So this was the first time I ever saw this happen. A bachelor male, Fuji on the left, challenged one of the dominant males, Royal, who's on the right, and about 2 hours later, the challenger had killed the leader, and he was now the new dominant male. Now, there's 3 different points about the takeovers that I'd like you to carry with you during the talk. First of all, most of the time, there isn't any warning, not for the leader male, not for the females. They don't know they're about to be targeted. Second, the takeover itself is really quick. Most of them last less than a day. And perhaps the most intriguing, in the weeks that follow the takeover, something very dramatic happens to the females in the group. So before the takeover, this group, just like any group of geladas, had females in every reproductive stage. So there's lactating females, there's pregnant females, cycling females, and there's even immature females, young females that had not yet reached maturity. Geladas aren't seasonal breeders, and so at any given time, most groups have females in every one of these stages. But a few days after the takeover, no matter what their reproductive stage was before, all the females developed signs of sexual fertility and they began mating with the new male. So geladas are just like most other circumpontine primates in that they produce these visible sexual swellings during fertile stages, and we call this cycling. So, but unlike other primates that have these sexual swellings, gelada sexual swellings show up as beaded vesicles around a bare patch of skin on their chest and neck. Now, in the adults, the vesicles are organized around this chest patch, but in newly cycling females, as you can see on the far right, the entire chest swells. So at the time when this happened, I remember having one thought and one thought only, which was, what an unbelievable coincidence. How did Fuji know that this was about to be a gold mine group where every female would return to cycling? But across the next decade and a half, really, I would continue to observe this same pattern again and again and again and again. And so a lot of my research program from that moment until now has really been motivated by these observations. So what is going on here? Okay, before I get to the research questions, I want to just step back a bit. I was a biology major in college, and in every hormones and behavior textbook on the planet, you can find a page that lists these effects. They're caused by pheromones in the urine of male mice. There's the Bruce effect. This causes a recently pregnant female to abort. There's the Vandenberg effect. This causes the onset of fertility in juvenile female mice. And then there's the Witten effect, and this causes the onset of fertility in adult females. There's others as well. There's the Lee boot. I could go on. I remember learning about these in an animal physiology course at Boston College, and I dutifully memorized these definitions, and I was able to regurgitate them on an exam. But what I distinctly remember is that the textbook really provided no context, no reasons for why these exist. And I remember thinking at the time that this must just be nothing more than males manipulating females to their own end. In each case, a female has to hurry up so the male can mate with her. But especially if we consider the first case, the Bruce effect, it's kind of hard to imagine why aborting a current pregnancy might be adaptive for a female mammal. So this brings us to a basic principle behind female reproductive strategies that's guided most of the research, especially for mammals. And that's this. The single most important difference between the sexes is the difference in their investment in offspring. The general rule is this: females do all of the investing, males do none of it. Now, we of course know Trivers' general rule has exceptions, and especially among the birds, we can find lots of these. But as guiding principle, especially in mammals, it accurately defines the primary source of conflict between the sexes. Most of the energy and time invested in offspring comes from the females. And it's from this basic fact that follows that male reproductive success is really typically limited by the number of females they mate with, and female reproductive success is limited by the time and the energy that they have to invest in each offspring to make sure that they survive and they reproduce. So these radically divergent interests with respect to reproduction creates a conflict of interest between the sexes. It sets up this inherent evolutionary arms race where many species, where each sex tries to one-up the other through evolutionary time. Perhaps the most effective weapon in the male arsenal for this arms race is sexually selected infanticide. So this is when males kill unrelated dependent infants that were sired by the previous male. And the idea is if a male kills an infant, this then accelerates the mother's return to a fertile state. This then allows the infanticidal male the opportunity to mate with her a lot sooner than if he hadn't committed infanticide. But anyone who studies sexual selection also knows females aren't defenseless. And often when we see infanticide by males, we also see one or more counter strategies by females. I'm organizing these female weapons, if you will, by the broad principle that allows them to escape infanticide. So one might be by confusing reproduction in some way. Females can either mate with many males to confuse paternity, and this is a very typical strategy for females that live in multi-male groups, or if it's a situation where there's really only one male and then there's an incoming male, females can confuse the male about their own fertility. They can signal fertility when they're really not. Second, females can accelerate their reproduction. Basically, they can hurry up. In these cases, females give up on investing in their current offspring, and they then move on to the next one. And finally, females can suppress reproduction altogether. In these cases, females suppress their own reproduction to wait for a better time. And this time might be ecologically a better time, maybe internally they're in better health, or socially it could be a better time. Okay, so back to these phenomena. If you're following the story, you probably already are starting to see a little bit more context for these than the one that the textbook provides. So are males simply just winning this battle of the sexes, or might females also stand to gain from these? And also, are these unrelated phenomena? Are they just three oddball effects? Or is there maybe some unifying principle that helps explain them all? I'll come back around to these questions at the very end. But with that overview and introduction, I'd like to start with my research questions. First of all, and essentially, they're what, how, and why. The what. What happens after male takeovers for females in each reproductive condition? I've just told you the story of Royal and Fuji, but going sort of beyond these anecdotes, are these changes real? And can we examine them statistically? Second, how is this happening? What's the mechanism behind these changes? And finally, why does female reproductive condition change following these takeovers? And by this, I really do mean, is it adaptive for the female? I don't care too much about the males. So my study subjects are a population of wild geladas living in the Simien Mountains National Park of Ethiopia. And we've been monitoring this population from 2006 until today. We're still managing some observations even now through the COVID pandemic with our Ethiopian team out there. And we're hoping soon to get back out. Now, across the years, we've followed anywhere from 15 to 20 polygynous one-male units. I put one male in quotes because there's often a subordinate, one or more subordinate follower males in the groups as well. But the dominant male does by far the most mating. And so the data I'm going to present to you today is kind of a mishmash. You know, some studies are earlier, some are later, but for the most part, you know, the biggest study represents more than 300 females across 48 male takeovers. Geladas live in a multi-level society. And so the society itself is comprised of a bunch of these one-male units. Each one-male unit has a dominant leader male, possibly one or more subordinate males, as I'd said, and anywhere between 1 and 12 adult females and their offspring. And these females are all related. Then at the periphery of the group, we have these all-male groups full of bachelors. And as I said earlier, these males are really just waiting for their time to challenge and defeat one of the dominant males. Now, generally, you don't find just single one-male units roving off alone. Dozens of these units typically overlap, both in their sleeping and ranging patterns, and this is something we call the band. And finally, even members across bands will occasionally overlap. And so any gelada group that comes in contact at some point across their lifetime, we compile as we call them part of a community. Okay, to understand the context of the questions I'm asking, you need to know just a few more things, mainly about male and female reproduction in geladas. First, the unit males do sire most or all of the offspring within the unit. Our genetic analyses detect no extra unit paternity. So within the unit, you get about 80% by the dominant and maybe 20% by the subordinate, but you never get sirings from outside the unit, from some male from a different group. Also, these takeovers happen about once every 2 to 4 years, but keep in mind there's a huge range of variation, minimum being a month, maximum being 7 years. And also, I'm sure you saw this coming, males kill infants. So in fact, male infanticide doesn't just occur, it's actually almost expected. It's really common. It actually accounts for more than half of all infant deaths. Okay, so what about female reproduction? 2 quick pieces. First, female geladas mature just before they hit about 5. Again, this age at maturation also is highly variable, but it's important to note that males at the long, so when you have males at the long end of the tenure, remember I said there's some about 7 years, they actually might have daughters who are about to mature or did mature in this group. And there'll be more on this in a bit. But you can see that there is potential overlap with the two ends. And second, the female interbirth interval. So the interbirth interval is the time from the birth of one successful infant to the birth of the next infant. And if the infant dies, we don't count that. So it's really a successful birth leading to the next one. And this is about 2.5 years. I'll come back to these dates in a bit, or these time periods in a bit. So a bit more about the methods. We have individually recognizable geladas. We know them by their faces. We monitor them daily or near daily, or as now, weekly, and we update their demography and behavior. We collect fecal hormone samples. So everything we do is non-invasive with respect to our hormone sampling. We just follow them around, and when they drop a fecal sample, we collect it. And then we later extract and measure steroid hormones for these individuals to go along with the behaviors that we marked down at the time. Okay, so let's start out with lactating females. What happens after male takeovers for them? Now remember, these are females who are nursing their infants. They're infertile until this infant is weaned. And the infant's usually weaned around 1.5 years. So what do we see after takeovers? You're about to see a bunch of figures that will look very similar to this one. So usually on the y-axis will be the number of females in a given reproductive stage. In this case, it's the number of females who've returned to sexual cycling. And on the x-axis, you're going to see days, weeks, or months before and after a takeover. And so the colored line will usually represent the takeover itself. And so you're looking at the frequencies of these observations on either side, with time 0 indicating the takeover itself. So it's pretty clear from this figure that it wasn't just the females in Fuji's group that suddenly wean their kids. After takeovers, as a rule, we see lactating females disproportionately resume sexual swellings. And so here we can zoom in on the post-takeover period only. You can see that it's really in this first month or two that there's this increase. And no surprise, when we compare these females to those who didn't have a takeover, we did find that lactating females who experienced a takeover resumed cycling much earlier than those that didn't. Okay, so females returned to fertility faster. But is it real fertility? Are they really fertile? We have evidence previously that in hamadryas baboons, females can exhibit deceptive sexual swellings. So we wanted to ask whether these were real or fake. So the mean interbirth interval for this population is about 2.4 or 2.37 years. So the question is, what do the interbirth intervals look like for the lactating females that showed signs of cycling after a takeover? These are our predictions. So if females really are exhibiting true fertility, so they wean their offspring early, then this is what we should see. Lactation's cut short, the other two stages remain the same, and this creates an overall shorter interbirth interval. But if the females aren't really fertile, so they're basically confusing fertility to the male and they're producing fake sexual swellings, then we should expect this second pattern. Again, lactation appears to be cut short, but for this prediction, the female doesn't get pregnant any sooner because she's not really fertile. She's just cycling for a lot longer. So it's really this bit in red here that's our primary focus, because under false fertility, a female appears to be cycling here, but she's actually still lactating. Okay, so when we look at the lactation period and we compare those with a takeover to those without a takeover, we find that indeed, following a takeover, the lactation period is shorter than the population mean. But that doesn't help us. You can see that lactation being cut short fits both pieces of the puzzle. We really need to now see how long are the interbirth intervals. Sure enough, when we compare the interbirth intervals for females before and after, with and without takeovers, we see that there's really no difference. The interbirth intervals are no different than the population mean. And so we get support for the second of these predictions, false fertility. So even though lactating females resumed cycling a lot earlier after takeovers, or at least they appeared to resume cycling a lot earlier, they didn't get pregnant any earlier. And we don't know if they're not ovulating, not conceiving, maybe they're aborting. We can't make a statement about at what stage, but we do know they're not getting pregnant. We do know they're not producing infants. And so this is contrary to what you might expect for the Witten effect. If males are actually prompting females to return to a cycling state in its true fertility, we would call that the Witten effect. And so I'm probably not going to mention the Witten effect anymore throughout this talk. So they're fooling the males. They're signaling fertility when in fact they're not. All right, let's take a look at pregnant females. Moving on to what happens after takeovers. So we tried to examine what was going on in pregnant females after takeovers, but there were no pregnant females after takeovers. So for this was based on the publication in 2012, and that was just a 5-year period. So this was a 5-year data set across 28 takeovers. But every year I update this data set. I'm just somehow obsessed with it. And even now, we only have a dozen or so females who have ever escaped the Bruce effect. And this is of hundreds and hundreds of pregnancies, and very few escape. And often there is some weirdo effect, like a female was off on walkabout, or, so eventually I'd like to have a large enough data set to understand these escapes. But for now, it is such a unifying principle. The males come in and everyone aborts. So for a non-seasonally breeding species, this is way different from what you'd expect by chance. Statistics are significant. And of course, the title of the manuscript is a bit of a spoiler here. We are seeing the Bruce effect in geladas. So let's dig out that textbook again. The Bruce effect is when a female terminates a pregnancy after exposure to a novel male. So there's no aggression needed. It's literally just the female gets a cue, there's a new male present, and she spontaneously aborts. The phenomenon was first discovered in house mice by Hilda Margaret Bruce in the 1950s. Then in subsequent decades, the Bruce effect was demonstrated in a bunch of other rodent species, but it could only be replicated under laboratory conditions. There were some reports of what appeared to be a Bruce effect in a handful of wild populations, but all of them were either indirect or anecdotal. So it was so much so that direct evidence of the Bruce effect, because direct evidence of the Bruce effect was so sparse in any wild population, people began to propose that it might be nothing more than a laboratory artifact. But these data that I just showed you from a wild population are also nothing more than indirect evidence for the Bruce effect. We're showing that there's loads of pregnancies prior to a takeover, and then after the takeover, there's a bunch missing. If we want to actually use direct evidence, we need to show that females were pregnant before the takeover, and then when the takeover happened, they were no longer pregnant. And of course, here's where our hormone sampling really pays off. So based on daily samples from a subset of females, and this is where Ayla's heroic efforts come in, because she was getting daily samples from a whole group of females, we were able to use estrogens data to show that a Bruce effect occurred not just after the takeover, but the same day as the takeover. So if you look here, these are 3 different females. These are just representative females. Fecal estrogens are on the y-axis, day relative to takeovers on the x-axis. And you can see that, this slow rise in estrogens signals pregnancy, just like you see in humans. And then the day of the takeover, they're no longer pregnant. I still actually get a little choked up about it. I mean, they're literally pregnant in the morning and they've aborted by the afternoon. This is far, we kind of went into this whole study expecting some form of Bruce effect, but the degree and the rapidity of how quickly it happens was nothing like we ever expected. All right, so moving on. Immature females, what's going on for them after takeovers? This is where we really do need the 15 years. So as I said before, we really did expect the Bruce effect a few years into our study. We had wondered about deceptive sexual swellings in geladas, and we wondered this forever because of the hamadryas example. But the process of maturing is this process that occurs in every single female. All females have different variables that affect it, might make it earlier, might make it later. So on top of all this variability, trying to identify whether males have an impact on this process took a long time coming. And these are long-lived animals. They don't mature until they're 5. So to gain the data set that we need has taken, I mean, we could actually wait longer if we wanted to, but I feel like it's time. So after takeovers, we did see that there's an uptick in maturations. We sort of added up the 3 months, but you can see you could probably just use the single month after a takeover. We used a logistic model, and we found that immature females were 3 times more likely to mature after a takeover. So let's go back to the textbook. The Vandenberg effect is when a novel male accelerates puberty in an immature female mouse. It was discovered by John Vandenberg in 1967. So is this the Vandenberg effect? I showed you that males stimulate maturations, but I didn't say whether they were early or late. So when we look at the timing of these maturations, we found that some were indeed early and others were indeed late, and some were on time. So takeovers in general tended to accelerate maturations. And so that's the dark green one and the dark blue one. You get a little push to the left. But when the female's father was present in the group, this tended to delay maturations. And so it would push everything a little to the right. So rather than a Vandenberg effect sensu stricto, and I'm saying that because I'm using the original definition of Vandenberg, at the end of the talk, I will propose a new definition. We'll refer to this bimodal process simply as male-mediated maturation. So to be agnostic, I'm just going to call it male-mediated maturation. Doesn't indicate whether it's early or late. And that this is really the unifying principle for these females. The male comes in, they mature. Some are late, some are early, but they're all male-mediated. Okay, so that's the what for the 3 different groups of females. The next bits are going to go a little faster because there's things that are more unifying across each one. How is this happening? I want to explore this a bit. Can we say anything about the mechanism? So we're somewhat restricted in what we can do with wild animals, but our non-invasive fecal hormone samples have given us some clues. So for instance, for lactating females, we can see that right after a takeover, there's a dramatic rise in estrogens. And this is well above baseline for lactating females. I mean, lactating females, if you look across their lactation, there is nothing going on. They're just, estrogens just stay flatlined. So the fact that we're getting this blip, this uptick, indicates something is going on. It's not that surprising because we know estrogens are integral to the process of sexual skin swelling. But what's weird is that it's lactating females. Like, where are they? Why are they suddenly generating all these estrogens to have these sexual swellings? Okay, what about pregnant females? Are their estrogens also going up? Well, here's where we run into some logistical headaches because the females aborted so quickly, we didn't have a big enough window to collect any samples between when they were pregnant, there was a takeover, and when they weren't pregnant, it was all the same day. And so we just don't have the resolution to get at that whatsoever. But we can look to other animal studies to see what's going on in those animals. Okay, so how do other animals spontaneously abort? Well, we know from a study that Matt actually led on yellow baboons that severe aggression from a male can be associated possibly with physical trauma to a pregnant female, and then this is often associated also with abortions as well. And this may overlap to some extent with the next one, generalized stress response. So under the generalized stress response, environmental challenges, in this case, maybe a male harassing a female, can cause a stress response. And the HPA activity associated with the stress response can then cause a female to suppress her reproduction. So we know a lot about how all sorts of stressors can suppress reproduction. And there's this general link between HPA activity, so that's the stress axis, and HPG suppression, and that's the reproductive axis. Now, whether this is what's happening in some cases of feticide is still a bit of an unknown, but for sure there's cases that, they're separate, 2 separate things to look at. Okay, so third, in the absence of any aggression or stress, a male can shut down female's reproduction using pheromones. So for example, in mice, it's been shown that the Bruce effect's mediated by estrogens, mainly estradiol. So research has shown that estradiol in the urine of a new male then enters the vomeral nasal organ of the pregnant female, and it can bind to uterine estrogen receptors in the pregnant female. Now, although estrogens are necessary for pregnancy, it's also been found that even slight elevations from the norm can actually disrupt pregnancy altogether. And so Denis de Catanzaro and many of his colleagues who do this research, they've injected radiolabeled estradiol into a male mouse. They then trace this estradiol from the male's urine to the female's vomeral nasal cavity, through her circulatory system, and finally to estrogen receptors all along her reproductive tract. But it actually even gets more interesting. So if pregnant females exposed to estradiol from a novel male abort their pregnancies, if immature females are exposed to estradiol from a novel male, they actually mature earlier than they would otherwise, exhibiting the Vandenberg effect. So we get the same kind of idea. Estrogen comes from the novel male's urine. It enters the vomeral nasal organ of the immature female, and it causes her, binds to her different reproductive tissues. They double in size and she matures. So it's been proposed that the Vandenberg effect and the Bruce effect may actually piggyback on the same mechanism. It may be some sort of pleiotropic effect, or at least that's what happens in mice. So Vandenberg and Bruce go hand in hand. When that paper came out, the 2012, that was sort of when the aha moment came, and we were thinking, all right, we got to really tackle this. Okay, so I've given you a sneak preview of immature females. Let's look at what we actually find in them after male takeovers. Voila, after takeovers, estrogens more than doubled for females, immature females, and not just 4-year-olds. I mean, we're talking 1-year-olds, 2-year-olds, 3-year-olds, and 4-year-olds, and all the way up. These are females that don't mature. There's no possibility that a 1 and a 2-year-old female is going to mature, but they still exhibit this rise in estrogens. And if we look longitudinally at different individual females, we can see the timing a little bit better. So the orange lines are the takeovers, and in some cases you'll see more than one takeover. And then you can see the rise in estrogens usually is within a few months from the takeover. So the first female all the way to the left is a 4-year-old. The next one in the middle is the 2-year-old. And the one all the way to the right is a 1-year-old. So as we see for mice, estrogens also seem to be playing a role in geladas. But remember that the estrogen in mice is coming from the male. So that's why I put this one, this pheromonal stimuli is sort of a rodent mechanism. And I don't think this is what's going on in geladas. So I'd just like to propose, before I move on to the why, I want to propose one more mechanism here that we know nothing about. And that's this sociocognitive mechanism. So catarrhine primates, like geladas, like baboons, they don't have the functioning structure, this vomeral nasal organ that processes chemosignals. And so there's a very high chance that chemosignals, which is the new term for what used to be called pheromones, don't operate in catarrhine primates. But additionally, I showed you some pictures of the geladas. They live in these huge herds, and they're often on top of one another. So the idea that they could actually target a chemosignal to a specific individual is also very unlikely. So I really do believe, I don't have evidence, concrete evidence for this, but I believe that it's more of a sociocognitive mechanism that causes females to then cause her own endogenous estrogens to rise. So they perceive this new male, her own endogenous estrogens rise, and this causes lactating females to swell, pregnant females maybe to abort. We don't know. I don't have any evidence for that. Immature females to mature. Now, this, of course, remains to be tested, all of them. But there's evidence that non-pheromonal stimuli can cause some of these effects, like Bruce effect, in horses and dogs. Okay, final question. Why is this happening? Why might these phenomena be adaptive for females? Okay, let's start with lactating females. So if females are using false fertility as a counter strategy to protect their infants from infanticide, does it work? do females who swell have fewer infanticides than females who don't? Now, at this stage, our sample size is small, and I'll explain a little bit more about that. But we did find this approximate association where infants were less likely to be killed if their mothers exhibited sexual swellings after a male takeover. So you can compare these 6 infants to these 19. However, in these analyses here, we haven't updated it yet, the females that swelled also tended to be the females with the oldest infants. So, and oldest infants are also less likely to be killed. So we have this confound. So we broke it up by age category. And when we look within these age categories, it's so disappointing. It's really clear there's no advantage to swelling. Rather than an effect of false fertility, we're more likely just picking up on the fact that older infants are less likely to be killed. So older infants less likely to be killed, also moms more likely to swell. It gets kind of difficult. What we need to do is get rid of these 2 outer categories. Get rid of the really young infants because those moms can't swell. Get rid of the really old infants because they're all at the advantage. They're all maybe outside the window of when infanticide is a danger. And we need to focus in on this middle group. We have to update. We have a few more years of data. But my guess is we're not going to have the story for another 15 years. No, hopefully another few years. Okay. So stay tuned. That's the moral of that story. Okay, what about Bruce effect? Is it adaptive for pregnant females to abort their fetus? Okay, well, here's the interbirth interval for our geladas. It's exactly the one I showed you before. It's right around 2.5. So, of course, when an infant is killed, her interbirth interval between surviving infants, remember, if an infant dies, you have to still wait for the next surviving infant. So her interbirth interval gets extended. It's much longer if the first infant dies. None of this is surprising. But what is surprising is when we compare the interbirth interval for the Bruce effect to the interbirth interval to females who lose an infant, but also to females whose infant lives. And you can see that the Bruce effect is actually not all that costly. They're able to turn it around really quickly. So in other words, losing a fetus now is probably better than losing an infant later on. Okay, so Bruce effect's maybe not that costly. Infanticide we know is pretty costly, but could there be other advantages? So we've started proposing this one for a while now. It seems to fit with so many of our other different data sets, our seasonality data, other things. It's what I'm calling the timing hypothesis. So this is the idea that all things being equal, female geladas should time the start of a new reproductive event. So cycling and mating with this new male. This new reproductive event should be timed with the arrival of a new dominant male. And this is because the average interbirth interval is just slightly shorter than the average male tenure for this species. So this means that each infant is more likely to be weaned the moment a new male arrives. So like the arriving male sires an infant, and right at the time that infant's about to be weaned is about when he's losing his steam and another male comes in. And so females might be able to take advantage of this timing to keep their infants alive. Okay, what about immature females? How's it adaptive for them to mature when a new male arrives? Now, at face value, the simplest explanation is it's a head start. It starts them off a little bit sooner, and indeed it does. So females with male-mediated maturation gained about a 4-month head start when it came to a successful first birth. But I mean, 4 months, it's a one-time deal across a long lifespan. It literally translates to like 0.14 offspring advantage across a lifetime. Is that really a sufficient gain for such a dramatic response? So the second thing we thought to look at was like, okay, well, what about the timing hypothesis? This could also be going on for the immature females. It's a little more difficult to assess because immature geladas have, right the moment they mature, they have about a 1.4 year period of what we call adolescent sterility. So they don't get pregnant for about a year, 1.4 years before they finally get pregnant. But we still were able to find some support for this hypothesis. So females with male-mediated maturation were able to buy themselves more time in order to get through sterility, cycling, gestating, and weaning before the next male takeover, compared to females who didn't mature, who didn't have a takeover-mediated maturation. So on average, the females who had male-mediated maturation were takeover-free for about 1,087 days. It's about 2.9 years. So basically their infant is about 1 year old by the time the new male comes in, which is exactly what gets their infant out of that danger zone. By contrast, the females on average who didn't have male-mediated maturation have about 727 days, about 2 years. And so this means their infant is actually right in the most vulnerable period, if, assuming all things being equal with the male takeovers. So cool, we got support for the timing hypothesis. However, does it translate to infant survival? The answer is not yet, no. I mean, the answer is no if we look at the data we have. Basically male-mediated puberty did not translate to higher survival for these females' first infants. But our sample size is super small. I think for takeover-related mortality, it was only 4 infants across 15 years. So the limiting factor here is a female with a known age. Okay, we need to know which of these females who have known ages mature after a takeover. Now, which of these females have at least 1 interbirth interval? And so just accumulating this kind of data set just takes forever. So again, stay tuned. But I want to throw one more hypothesis out there. This is one that was first raised by Thorpe and de Catanzaro in 2012 when they reported on the kind of going together of the Bruce effect and the Vandenberg effect. I'm calling it the byproduct hypothesis. So this is the idea that female sensitivity to this arrival of novel males might just exist as a byproduct of the adaptive Bruce effect. So the Bruce effect is adaptive in geladas. It's thought to be adaptive in other taxa where it exists as well. And the two are mediated by the same mechanism, at least we know that in mice. So as long as it's not overly costly, it's possible that the Vandenberg effect might simply just piggyback on the Bruce effect. Now, this one's difficult to test at present, but I can offer some tentative support for this. I did a comprehensive literature review of all confirmed cases of Vandenberg effect and all cases of Bruce effect. And in every single case, to my knowledge, every single case where Vandenberg effect occurs, so does the Bruce effect. All 10 of them. I mean, it's only 10, but that's what we got. So testing this hypothesis properly does have to wait for a lot more data from a lot more taxa before we can do actually a proper comparative analysis. But that's pretty exciting. All right, so to sum things up, what happens after male takeovers? Well, females show immediate signs of fertility. Sometimes it's true, sometimes it's false. How is this happening? The mechanism appears to parallel what we see in mice, with this rise in estrogens in the 2 cases where we can actually get in there to look, the Vandenberg and deceptive swellings. Okay, but why is this happening? Is it adaptive for females? Well, for lactating females, false fertility is associated with a decrease in infanticide, but at present it's mixed up with infant age. So we need a little bit more data, well, a lot more data to sort this out. So stay tuned. For pregnant females, the Bruce effect prevents females from continuing to invest in a doomed offspring. It also sets them up, the timing hypothesis, it sets them up to wean the infant right around the time of the next takeover. And finally, for immature females, the Vandenberg effect may ultimately prove to set females up for success with the timing hypothesis. But for now, we've also have this viable non-adaptive hypothesis, and these are overlapping, which could very much be at work here, which is that it's piggybacking on the Bruce effect. Okay, so to wrap up, I want to return to these 3 phenomena, especially the first 2, since those are the ones that seem to be most relevant for geladas. So even today in 2020, I teach hormones and behavior. I still have yet to find a hormones and behavior textbook that gives any evolutionary context to these effects. But when we're thrown into the field with animals, we're observing animals day to day, and we come really face to face with how real these phenomena are. Some of them were obvious. We knew in our first year, geladas probably had the Bruce effect. Some took a lot longer to appreciate. I don't think I fully believed in Vandenberg until maybe last year. And then some are just clearly not present, like the Witten effect, since the return to fertility was just a false one for geladas. But my 15 years with geladas has also led me to extend my musings on these phenomena. So if you'll indulge me, I'm going to put up this photo of me. It was just taken a few months ago, almost exactly 15 years later from the photo that I showed you when I opened the talk. So now you can imagine me musing about these phenomena in geladas. So for example, I don't think the Vandenberg effect is a real thing. I think it's just male-mediated maturation. I don't think it matters when this happens. If it's the same signal, and assuming it's the same neuroendocrine process, why do we need a different name for a phenomenon that accelerates maturity versus one that just triggers it? And so even though in geladas, there's this male-mediated maturation, sometimes accelerates, sometimes delays, I mean, sometimes it's too late, I would push that all in the same category, calling it Vandenberg, or let's just call it something different altogether, male-mediated maturation. I'd also argue that all these phenomena are connected in some way. So where males are definitely this strong selective force on female reproduction because they commit infanticide, there's also going to be strong selection for females to respond to this reliable cue. All females respond with this surge in estrogens. So it seems like if you're a really young, immature female, it does nothing. Your estrogens rise, it goes back down, you're back to normal. For slightly older immature females, it triggers maturation. For pregnant females, regardless of what stage of pregnancy you're in, it triggers abortion. For lactating females with very young infants, it might not be enough to trigger sexual swellings, but for females with older infants, it triggers these sexual swellings, but not yet enough for ovulation. And then for females with much older infants, maybe this actually triggers a true return to fertility. As long as it's not costly, all of these things will continue to persist. So to end this talk the same way that I feel like I end all my talks these days, we just need more data from more taxa. So the mechanism has to be more fully examined in a captive study, a captive setting, but the function, really to understand the evolutionary function, we need data from numerous other closely related as well as distantly related taxa so that we can do a proper comparative analyses. And with that, I'd like to thank everyone for listening, and I'm happy to take questions.
Thank you so much for that, Jacinta. I thought that was a really wonderful talk.
Thank you.
It looks like we have 6 questions now, while rapidly accelerating number of questions, and I have a couple too. So we'll start with Ann Pusey, who asks, she says, do juvenile females that mature early because of a takeover experience earlier first birth, or is their early cycling deceptive? I think you mostly answered that, but I'm wondering if any of that earliest cycling is deceptive.
Good question. So from what we understand, so when a female cycles early, it does take her longer than the population average to get pregnant. So there is something going on there, and we are exploring that further with growth, size, a lot of different things with life history. But they seem to still catch up. I mean, they actually surpass females in the sense that they wean them faster, I think. Sorry, I'm getting that mixed up. They do get this 4-month head start. So they end up having their first birth sooner. It must be that they cycle for longer, but they still don't cycle as long as the ones that mature after without a takeover.
That makes sense. Okay, Lisa Danish asks, will a female who experiences a takeover early in her life, say 1 or 2 years old, then begin her reproductive career earlier several years later, such that does the estrogen rise in the 1 and 2-year-olds have an effect later in
Yes.
life?
That's a great question, and we had it right away. I remember thinking like, oh, well, maybe it primes them. It's getting them pumped up and they will actually mature sooner. The answer is no, they don't. We looked at the number of takeovers. We looked at the estrogen itself. There's nothing, a female that has a takeover at 1 isn't more likely to mature sooner than a female who doesn't have that. And this is actually removing the effect of the 0 takeover female. So they skew everything because 0 takeovers means dad is your leader. So if you remove, and those of course are late. So we remove them before we do these analyses.
Great. Mark Halber asks, why do you think that humans and other primates don't have a dedicated vomeronasal organ when so many other mammals do?
That is a very good question. So this is my musings. So it does seem like there's a divide at catarrhine primates. So there is a vomeral nasal organ in lemurs, lorises, I believe tarsiers, and New World monkeys. There's some maybe evidence that it exists in mandrills, and there may be a few other catarrhine primates that kind of have weird scent marking kinds of behaviors. So it's not entirely clear cut, but for the most part, catarrhines, we are missing the holes in the top of our mouth where it gets pumped into the vomeral nasal organ. And all of our sense of smell comes from volatiles, comes from our main accessory olfactory organ. Why? I actually think this is in line with a lot of these processes that used to be guided or in some sense dictated by hormones. So we've sort of, I think primates in general have emancipated themselves. I think that's a term Kim Wallman used, but emancipated themselves from hormonal control. And partly because smell doesn't play as large a role in our lives, but also because sociocognitive or cognitive processing has taken up so much of a role in our lives. And so we take in our stimuli in different ways. And we're processing everything visually, but then we're processing what it means. And so I think this, kind of emphasis on understanding what's going on around you is more important than, accidentally getting a urinary smell. So if you imagine the geladas, they're just piles of fur, where they don't even know each other. So one female might have her back up against the male of another group. She might put her hand in his urine. I could see all kinds of disasters if suddenly pheromones took over and we're back in control again. Whereas, a female sitting there knows that's not a viable mate. Like she's not going to mate with that male. Whereas she understands when her dominant male has been replaced with a new dominant male. And I'm using understands in quotes, but there's probably a lot more sociocognitive processing that then triggers the same downstream neuroendocrine response, just guided up here by a different stimulus.
Lilia Badescu asks, have you looked at what generally happens to infants once a takeover happens and lactation is cut short abruptly? I expect that it would depend on infant age or infant stage of nutritional development, but if the takeover phenomenon happens so frequently in this species, maybe we would expect a lot of infants would deal surprisingly well with abrupt weaning. So what kind of survival outcomes do you see in those abruptly weaned?
Right. So it's a really good question. It's not one that we've been able to really get at. Part of the problem is the coarseness of our data. And so, what we see, whether or not a female is abruptly weaning, we call it an abrupt weaning if the infant lives, because she's now starting to swell again and the infant lives. But often if the infant dies, we don't know necessarily why, whether it's infanticide or not. And so some of it is just the coarseness of our data. And again, that's one of those problems where we just need to accumulate more data where we're confident of those windows to really assess it. But I don't have a definitive answer. There isn't any obvious pattern in the sense that, oh yeah, these females who wean early, they always lose their infant. I'd say more often than not, it's the opposite, but I haven't done a real in-depth analysis on that yet.
Eli Strauss asks, he says that it looks like there's a small peak in estrogens after abortion in females undergoing the Bruce effect from those 3 plots, estrogen plots that you showed of the 3 females. Was that a robust pattern? And do you have some idea of what might be going on there?
Let me try and pull those up. So I'm not sure. Oh, this little pattern here.
I think that's right.
Aha, okay. So these are days relative to takeover. And so that's probably the next sexual swelling, the start of it.
So they go right back into swelling.
Oh gosh, days. It's like, it's crazy. That's why I think, you know, that this image here is as, sorry, trying to find it. Can't find my own slides. The Y part here. That's why these two are so similar, is because yes, they abort. So they lose whatever months they've gone into pregnancy, but they turn it around right away and they get pregnant. So it's definitely fertile. Now, of course, this compiles females who lost it in the 1st month, 2nd month, 3rd month, 4th month. And there's very few, and we have a few in the 6th month who also lost their fetus. So we know it can happen, but my guess is it's less common.
The next question from Buxter and Rudy is, not sure if I missed this, but do you think that the age of the female or the number of times she has successfully raised an infant influences whether or not she displays false fertility and swellings following subsequent takeovers?
That's a great question too. We have pushed at that and there is no signal. We also looked at whether, you know, we've looked at, sorry, no. The analysis we did was whether their infant lives or dies. We have not done that analysis on whether a female swells or doesn't swell. And the reason, yeah, no, that's a great point. So looking at sort of what causes females to swell and not swell is not something we've pushed, but it's a good suggestion.
Shahid Gashi asks, does the reason of abortion happening with the Bruce effect, is that so that the females can mate again and then the new dominant male will take care of the offspring because they're his offspring? Or do you think there's any other ultimate explanation?
So the males do no care, zero. Like geladas just do not have paternal care. It's so he doesn't kill the offspring. So and in fact, you know, I showed you this picture. Let me go back to it. This one here. So there's 2 females that swelled. When I published this bit, I knew about these 2 females. I was all over them. Now we have about 12 or 13. And so I haven't kept track of each one. I've looked at some of them. But I can tell you that one of these swelled. So she's a pregnant female who sexually swelled. The male mate mated with her. And one of her son is now a leader male in another group. Like he grew up, he was totally successful. The other female didn't swell, didn't mate with the male. The moment her kid was born, the leader males killed it. So anecdote, but it first of all, it does suggest that pregnant females also can swell. It suggests that there probably is, I didn't have hormones from this female, but it suggests that since we know estrogens go along with sexual swellings, it at least provides evidence that her estrogens probably are going up, even though she's pregnant. Yeah.
Okay. Pankuri Singhal asks, you mentioned that the duration between takeovers can vary, with the longest period being 7 years. Looking at immature females with these long durations, since their father is probably the unit male, do they show some form of reproductive strategy to avoid mating with him? And if the answer is yes, what is the extent of their kind of reproductive delay? Is there a limit?
So the strategy they use is to not cycle. So we definitely see these females, they don't, the male won't mate with them unless they have sexual swellings. in that case where we had the 7-year male, he was there for 7 years. He had 2 daughters and they just swelled, like they were already close to 6.5. I think, I don't know if I have them on, but they're always the extreme points all the way out on the chart for females swelling. They mated with their father. So if they if they finally mature, there's nothing that prevents them from mating with their father. They do behave differently. Like we did have another case where females did come in, they swelled, even though their father was the leader male. He was taken over within weeks. So it was a really quick little thing. But during that little time, they would actually, for the first time ever, we saw females within one group solicit, or present themselves to males from a different group.
And then I think here's the last question. Rachel Nelson asks, do all males have estradiol in their urine? And is there an increase in this when a male takes over? So what do we know about that in rodents and in primates?
Right. Yeah, the rodent literature is really quite fascinating. So males, if you're actually the leader male, sorry, I'm using the wrong terms. If you're actually the sire of the mouse and there's a pregnant female nearby, you have almost very little estradiol in your urine. So there, I don't know what the mechanism is, but they don't find a ton of estradiol in those males' urine. Also, we know, this is just a little aside, there are identifying urinary proteins in the urine. And so when the female is sort of forced to take in a male's urine who's the sire, she blocks the Bruce effect. She actually does not have Bruce effect because of this recognition component. So that's a little aside. Related to the new male coming in, his estrogen level in his urine is through the roof, exponentially higher. Not only that, but he urinates constantly and he actually wafts it towards the female. And the female has motivation to investigate the urine in a lot, like she investigates it constantly. And that usually when you don't recognize an animal, animals will investigate a lot for a lot longer. And so you see that. So that increases the likelihood that she's taking in that estradiol. Male humans have loads and loads of estrogen. If you wipe their armpit and then you basically test that swab. And so there's lots of possibilities. People are so fascinated by the idea of human pheromones. I don't buy it one bit. No way. I mean, I think we can smell something and find attraction in it. If you call that a pheromone, fine. But if it's like the pheromone that enters another human and binds to their receptors, no, I don't think so at all.
And then I had one question. So following male takeover, there's presumably some females who were cycling at the time of takeover.
Yeah, you notice I just ignored them.
Well, so my question is, do those females have fewer cycles following male takeover than they normally do in the population average?
That's a really good question. That's a good question. I don't know the answer. I've just started looking at their estrogen levels, but it's hard when they're cycling because they're up and down all the time. And you need a pretty fine-grained pattern from one female to really get at that, which I don't know that we have right now. But that is a nice, almost indirect way to look at it. So you kind of can just see if when there's a takeover, so normally it's 1 to 3 cycles. So then we would, that's the problem. There's not a ton of variability in there, but there's still a little bit more extremes in that, but there could be a signal. I'm going to look at that.
Well, thanks for giving this talk. I think this was really, really great. And it's terrific to see these kind of different branches all sewn together. So thanks for giving the talk.
Sure, it was a pleasure. Thanks to everyone for the questions, really spot on. And thanks for being here.
And for the folks out there who will be here next week, we have another talk about primates, this from Fernando Campos. I have the title. It's The Biodemography of Health and Fitness in Changing Environments, Insights from Wild Primates. So come back next week, same time and place to hear from Fernando Campos. Thanks, Jacinta.